After having studied cell biology, biochemistry, and molecular biology, and gaining a PhD in neuroscience from St Andrews, Dr. Berger joined Professor Gunmore's group, which has a key interest in Alzheimer's disease research. With a worldwide aging population due to improved healthcare, the importance of combating Alzheimer's is increasing. The chance of contracting Alzheimer's increases with age. 0.125% of 30 to 59-year-olds are sufferers, and this rises to 33.79% in the 90 to 95 age bracket. Approximately 815,000 people in the UK suffer from Alzheimer's. This number is expected to reach 1.7 million by 2051. Worldwide, the cost of caring for those suffering from Alzheimer's is £100 million and 23 million in the UK alone. In a patient suffering from Alzheimer's, beta-amyloid plaques and neurofibrillary tangles begin to develop in the brain. Beta-amyloid is generated by amyloid precursor protein, or APP, which is normally found on the surface of brain cells. APP undergoes beta-secretase cleavage and results in the amyloid proteins, which due to their chemistry stick together to form plaques. Small pieces of plaque can prevent cell-to-cell signaling synapses and activate immune responses, and larger plaques accumulate on brain cells and can lead to cell death. Neurofibrillary fibers are tangles of insoluble fiber. They result in abnormal tau protein that causes the microtubules to collapse and fall apart. This prevents nutrients from reaching the brain cells, which combined with the effects of the amyloid plaques leads to neurodegeneration. Much of Dr. Berger's current research involves the chemicals endophyllin-1 and amyloid beta binding alcohol dehydrogenase, or ABAD. She focuses on the effects of upregulation of endophyllin-1 on neurons in patients with dementia. This leads to how the pathways that cause oxidative stress in the brain, leading to neurodegeneration, are stimulated. Ultimately, they are trying to deduce what exactly is occurring at neural synapses that dampens their function and ultimately degrades them in early Alzheimer's pathology. The lab has also developed a treatment using decoy peptides containing a modified peptide sequence from ABAD, which slowed and reversed pathophysiology of dementia in mice. Early diagnosis of Alzheimer's disease is important to further treatment of patients. Unfortunately, currently used methods, such as psychological tests, detect the disease in relatively late stages. However, biochemical differences in intracellular mechanisms underlying dementia could be the key to developing new methods for early diagnosis. It is possible that changes in ABAD and endophyllin could be biochemical markers, unique to Alzheimer's disease. In that case, they could be key to both treatment and diagnosis. This is why in future, Dr. Berger would like to extend her work to investigating biochemical markers for other types of dementia. And even though these discoveries would only be baby steps towards the cure, one day they will take us to the moment when Alzheimer's disease will be just a bad memory in human history.